So hello everyone. Am I audible and my slides are visible?
Yes, ma'am.
Okay. So let's start the session. We have to just revise whatever we have studied in the previous days. So we have read the gut microorganism composition, their role in human metabolism, how they contribute in different kind of diseases, which is known as the dysbiosis. Dysbiosis. Now, the major medical prospects in this gut microorganism or the microbiome is that how it gets destroyed or disturbed through different environmental factors, and then what are the different consequences. Because you know about the type 2 diabetes, which is a metabolic disease, and India is known as the diabetic capital. So how this microbiome results in the type 2 diabetes? Obviously, the type 2 diabetes patients have an altered gut microbiome because all the food habits or the lifestyle habits contributed into the disruption of the normal gut microbiota of the individual. So how they just how the disease progression occurs, how the microbiome disrupts. So in this figure, you can see if you are consuming some, uh, if you are having some good food habits, or if you are consuming healthy diet or a balanced diet, then obviously your gut microbiome will be healthy. But if you are not consuming and your lifestyle is also not so healthy, then the microorganism of your microorganism will somehow be altered, and then this may lead to disease progression. If you are consuming an healthy diet, and also if you are having a very balanced lifestyle, like you are doing exercise, you are not taking stress, you are having a very fresh and clean food, then obviously your microorganism or the microbial metabolite will lead to a healthy lifestyle or a healthy life. But if you are somehow altering your microorganisms environment, then you are pushing them to be an antagonist of your life, and then they will just result in, or the consequences will be so many different kind of metabolic diseases. So the emerging evidences indicates that it is not only the microorganism and their structural component, but also their metabolites that influences the host and contributes to the development of the metabolic syndrome and type 2 diabetes. So I can give an example with my research. So microorganism, whenever we grow microorganism in a media, in my case, I use normal nutrient broth. This is the universal media which helps the bacteria to grow in it. This is the very basic and the universal media used everywhere for the culturing of any kind of bacteria. If you have isolated any kind of bacteria and you don't know about the selective media, just go for the NB. NB supports the growth of each and every bacteria. So I work with Pseudomonas, Pseudomonas aeruginosa, which is the very toxic bacteria, and it is a priority pathogen because this results in so many different kind of diseases, because it is a very potent toxic bacteria. It is known to cause so many different kind of diseases through its biofilm. So this is an antagonist for everyone. This is an opportunistic pathogen. The uh, different medical equipments like the catheters or the different even inorganic surfaces are infected by the pseudomonas. So this is an antagonist. But in my research, I am using this as a protagonist. For everyone, this pseudomonas, which is a gram negative bacterium, is a villain. But for me, this organism is a hero. Why? Because I am using its biofilm for the remediation of heavy metals. My work is related to molecular microbiology. So I am dealing with what genes are responsible for this in the pseudomonas that can uptake this heavy metal. So what I am trying to explain through this is we should know about the metabolites secreted by the bacteria. Whenever you grow a bacteria in a media, then the bacteria will show its metabolic machinery. It will start its metabolic machinery, then it will synthesize some metabolites which are very much useful. In the form of metabolite, they can synthesize enzyme, they can produce antibiotics, some proteinaceous molecule like bacteriocin and other metabolites as well. So all those metabolites are of great medical importance. So we have to find out that which metabolite is secreted by which bacteria and under which condition of pH, temperature, or any other stress. Like whenever I give a heavy metal stress, metal is a kind of stress. Nobody wants to live in a stress condition. So how the pseudomonas cope up with the stress condition? It synthesizes a biofilm. Biofilm is a organized microorganism with its extrapolymeric substances. So they just hide in the biofilm to prevent the ill effects of heavy metal. So likewise, if you are providing, this is your gut microbiome, all these are microorganism. So if you are repeatedly providing a bad environment for your gut, then this microorganism will synthesize some toxic metabolites that will otherwise harm your body. If you are providing this with a very good amount of nutrient, a sufficient amount of nutrient, and also a very good condition of environment of like temperature, pH, and everything, no toxins are produced in your body, then this bacteria will synthesize the good metabolites, not a toxin. So all it depends on how you are feeding your body, how you are providing the different conditions to behave the bacteria in a good way, like to be an hero or to be an antagonist. So all it depends on what kind of environment you are providing. So if you are providing a healthy condition, then the microbial metabolites will help you to uh, prevent different kind of infection. But if you are helping them, if you're not helping them to grow properly, then they will be converted into some toxic metabolites that will ultimately results in the disease promotion. Now the gut microbiome in humans can be considered as an organ which has function critical for human metabolism. If you can imagine that there will be no microbiome, if microbiome will be absent from our body, then so many different things will happen to us. What it may results in? It may result in low metabolism, because as I have mentioned yesterday that we always feed our body with complex polymers, but our cells cannot sense those polymers, so it can only sense the monomers. So for the conversion of polymers to monomers, this does not happen de novo. They require some hydrolytic enzymes. For example, proteins can be broken down by proteases, and carbohydrate can be broken down through amylases into ultimately glucose into amino acids. And also for the fat degradation or for the fat hydrolysis, we need lipase. So all those hydrolytic enzymes we need, and all those hydrolytic enzymes are the product of microorganism. So if microbiome is absent in a human body, then the metabolism will be very low. He or she will not be able to uh, digest the normal nutrient also, the carbs, the protein, the fat. So low metabolism, no energy, he or she will feel lethargic, digestion will not be in a very good manner, so this will also be hindered. Now, maintenance of gut barrier function and immunomodulation. No immunity will be provided. If no microbiome is there, no immunity will be provided. Now, the human gut possesses millions of microbes, as you know. So, there are four categories which we have studied yesterday that define a complex microbial community. We have also read that the microbiome similarity, if you compare microbiome similarity, if you will compare human 1 with human 2, then there will be a great similarity. But if you will compare human with mice or any animal, cow, goat, sheep, then there will be a dissimilarity, because species to species similarity we can see, but not with the species and genus. So the gut microbiota has been characterized as a vital organ, forming its multi-directional connecting axis with other organ. So this point is very important, and I will explain this with an help of a diagram. Many researchers have designated this microbiome as not just only microbiome but a vital organ of a human body, because they are uh, offering so many important roles. Without them we cannot even think of sustaining a life, because they help in metabolism also, they are preventing us from illness also, they are preventing us from so many infections, they are able to maintain the gut brain axis also, yani they are protecting us from neurodegenerative diseases. So they have a vast variety of roles, so we can consider them as much important as our heart, brain. So the gut microbiota axis is responsible for host microbe interaction and works by communicating with the neural, endocrinal, humoral, immunological and metabolic pathway. Obviously, it neural, how it controls the neural through the gut brain axis, endocrinal through different hormones. It can increase or either decrease the synthesis of the hormone by the endocrine glands. Humoral means blood related, immunological and different metabolic pathway, as it synthesizes short chain fatty acid, volatile fatty acid. So ultimately participating in different metabolic pathways. I will show you one diagram. This is the diagram of a gut microbiota strain. So first we will see this slide and then we will come to this. So the human gut microorganism are mostly non-pathogenic. So when this non-pathogenic will convert into pathogenic, when dysbiosis will occur. Dysbiosis means imbalance. If through different factor of age factor or environmental factor have deteriorated your normal microflora or your normal microbiome of your body, then this non-pathogenic strain will converted into pathogenic strain. And in our normal experiment also, if we keep bacteria uh, without reviving it for so many months, then obviously the chromosome of the bacteria will be altered. Let's suppose if you are having 100 culture, not culture, it will be colony, 100 colonies. Suppose this is a petri dish and you are having 100 colonies of bacillus cereus, right? You are having 100 colonies of b cereus. Then if you are expecting that all the uh, colonies are having this much of nucleotide in their chromosome, so you are wrong, because out of 100, let's suppose 99 will have same genome structure, but 0.1% there may be chances that its genome is altered. Somehow it is altered due to different environmental factor, like contamination may occur over time. So you can think that this non-pathogenic strain can be anytime converted into pathogenic strain if different sources are there, like contamination chances. In case of microbiome, how it can be converted? It can be converted through environmental factor, age factor, disease factor. All these things will promote this transition, non-pathogenic to pathogenic transition. So whenever you are keeping or working with 100 colonies at a time, or let's say 100 strains at a time, so you can't say with 100% surety that all these strains have having the same set of genome you have started earlier, because you don't know at what time its genome will be altered. So in my case also, whenever I do sequencing, I see some of the base pairs altered. Like if I in the first sequence result I got ATGC, then in the second I got AATG. Of course, this is very what to say, this is very non-specific, but we this is very minor change, so we do not take into our consideration that it is a major change. But of course, with time, there is an alteration in the base pairs of the microorganism, because they show different tendency. We are keeping it in different metabolic condition, like we are providing it some nutrient, many a times we change the nutrient condition. So after having so many changes, the bacteria show transition in its whole genome. So here also, the non-pathogenic will be converted into pathogenic. So how it will be converted? Through dysbiosis. So dysbiosis of the gut microbiota is therefore linked to various human diseases such as anxiety, depression, hypertension, CVD, obesity, diabetes, all those diseases which are caused due to the metabolic shift, metabolic changes. And also, the major form is cancer. The fatal form or the deadly form is the cancer, because in the cancer you can't control the growth of the cell. So the mechanism leading to the disease development has a crucial correlation with gut microbiota, metabolic products, and host immune responses in human. The subject, that is the gut microbiome, and its application has a medical importance, direct medical importance, because nowadays if any patient visits to the doctor or any hospital, then he or she may be advised to do the microbiome test nowadays. Microbiome test means they will go through different stool test, that what is the percentage of good bacteria and the bad bacteria in its fecal matter. If it is, if the good bacteria are higher in number, then its gut microbiome composition is normal. But if we are getting bad bacteria high in number, then its gut microbiome is somehow altered. And because of that alteration, he or she may be having some sort of metabolic illness. So all these things have a medical importance. So this is an overall summary of whatever we are having in our microbiome. Like if we are saying the talking about the stomach, then it has 10 to the power 1 to 10 to the power 3 CFU per ml of lactobacillus, streptococcus, staphylococcus, and enterobacteriaceae. So all these bacteria are common also in so many organs, and also they are specific also. So they are responsible for so many control and coordination thing, like they maintain the gut brain axis, they maintain the gut brain endocrine axis, and through the endocrine they mediate the hormonal regulation, hormonal regulation, gut heart axis, and through that they control the CVD, cardiovascular diseases, atherosclerosis, thrombotic events, hypertension, like low BP, high BP thing. Gut lung axis, it means pulmonary diseases. Gut liver axis, liver inflammation, liver cirrhosis thing. Gut pancreas axis. Now pancreas is related to what? The insulin production through the beta Langerhans cells. So Ultimately, it will control the diabetes because insulin is related to the diabetes. Gut bone axis, if it is altered, osteoporosis may occur, pores in the bones, pores in the bone may occur, and gut muscle axis. So sarcopenia, gut skin axis, gut reproductive axis, gut kidney axis, gut bladder axis. Now this will control your all kidney related thing. So see, this diagram shows the importance of what? The importance of gut microbiota. If the normal gut microbiota is somehow altered, if I am saying somehow altered means you should know about the factor responsible for its alteration. So if your gut microbiota is somehow altered, then you can have this thing, this type of diseases. If any of the diseases like gut, if your gut microbiota is altered, you can have the disturbance or the disruption in the normal gut brain axis maintenance. So it may result in stress, anxiety, depression, and also gut brain endocrine axis. Then hormonal regulation will be altered. Either one of the hormone will be in very excess or in very low amount, like the thyroid disease, hypothyroidism, hyperthyroidism. So all these things are under the control of gut microbiome. So before having this class, before reading this slide, we can't even imagine the this much role of a gut microbiome microorganism. So you can say that the microorganism inside the microbiome is providing with so many things to us. If they are not present, or if they are altered, if they are disrupted, then it is known as the dysbiosis. And you can say dysbiosis of gut microbiota can result in so many different kind of disruption of the normal metabolic machinery of a human body. And in an example, you can give any of the above example. Like you can see every organ is influenced if your microbiome is influenced. Now, I was reading one article that Hippocrates once said that everything is in your bowl. Bowl means the normal gut thing. So he once upon a time regarded in his book that the gut, if your gut is healthy, then you are healthy. If your gut is not healthy, then nothing can stop you to cause illness. So he have written one sentence, I will share the article with you, that everything is in the bowl of our body. Bowl means the gut. So now you can relate why he said this point, the father of medicine, Hippocrates. So you can see if your gut microbiota is disturbed, you can see different kind of illness. Now let's count the organ which are influenced. So the brain is influenced, heart is influenced, lungs are influenced, pancreas, liver, bone, muscle, skin, everything. Even you can see gut reproductive axis. Nobody can imagine, uh, like nobody can imagine that even if your gut microorganisms are altered somehow, this may results to reproductive illness also. Yes, it is possible. Infertility, PCOS, PCOD, ovarian dysfunction, everything is. And also after the menopause also, the post menopausal osteoporosis, the estrogen disturbance. So, so everything is the result of the gut microorganism. So how important this unit is to study. So this is related to the dysbiosis of gut microbiota, that what it can cause. Now next part. Now answer me, which of the following metabolic function is not significantly influenced by the human gut microbiota? Synthesis of short chain fatty acids, vitamin K production, digestion of dietary fiber, oxygen transport by hemoglobin.
Oxygen transport.
Oxygen trans.
Okay, अब कर दिया जाए ऑक्सीजन ट्रांसपोर्ट. You are right. Oxygen transport is not directly linked to the human gut microbiota. Human gut microbiota directly controls the synthesis of SCFA, that is short chain fatty acid, vitamin K production, because this is the function of the major contribution of the gut microbiota, the synthesis of vitamin K. And also, they are involved in the digestion of dietary fiber like carbs. They are helping us to convert it into monomers, but they are not directly controlling the oxygen transport by the hemoglobin. So this is the answer. So through this question, you get to know the exact three function of gut microbiome. Now next question. The major products of bacterial fermentation of dietary fibers in the colon are
Short chain fatty acids.
SCFA, short chain fatty acids.
yes, short chain fatty acids. So this was the reason of that we are producing short chain fatty acid by the conversion of the complex polysaccharide. So SCFA like acetate, propionate and butyrate are produced by microbial fermentation of indigestible carbohydrate in the colon. Now next part. So this diagram shows the positive health outcomes of a gut microbial species. In this slide we have studied what if the normal microbiota is altered, and in one word what can we say, the dysbiosis is nothing but the altered or the disrupted microbiome. One word for this is dysbiosis. Now we have seen that if our microbiome is disturbed or deteriorated through different factors, then this much of illness we can expect. Now this is the opposite of that, the positive health outcomes of a gut microbial species. So obviously it will be same, you can say they maintain the gut brain endocrine axis, they maintain the gut liver axis, gut pancreas axis, gut skin axis, and through how they increase the synthesis of SCFA, they increase the synthesis of antioxidant, so to kill the reactive oxygen species, because nowadays so many diseases are only because of the production of reactive oxygen species. So many kind of cancer also, and a different syndrome also, they just arise from a single reactive oxygen species. In our case also, the bacteria structure, overall structure of the bacteria is altered because of the ROS production. In my case also, the metal induces the production of reactive oxygen species, and then this reactive oxygen species needs a scavenger, antioxidant scavenger. So in our body, the antioxidant production is high, then no ROS will be there and no disease will be there. ROS also cause autoimmunity diseases. No inflammatory mediators will be transferred in our body if we are maintaining healthy gut microbiome. No pathogenic colonization, no pathogen will be there in our body. Improved lipid metabolism, no lipid storage, no fat storage for a longer time. Healthy gut epithelium will be there. If a gut epithelium is healthy, then what you can expect, you can expect a very good amount of and in very balanced amount of metabolites that will ease the process of digestion. Gut inflammation will be low, insulin sensitivity will be high, because in type 2 diabetes insulin sensitivity is lost, so it will be increased. It will facilitate the digestion process, vitamin production, beneficial metabolites, this I was talking about. It will not secrete toxic substances. Toxic substances will be no longer secreted if you are maintaining a positive and a very good gut microbial diversity. Immune response
Ma'am,
regulation
when
will
you
be
say toxic substances, so exactly what kind of toxic substances are made in the gut if it is disturbed?
If it is disturbed, they synthesize so many different kind of proteinaceous molecule also, so many different kind of carbohydrate molecule also, lipopolysaccharide, that may otherwise alter the overall bacterial species there if they are producing some kind of uh, carbohydrate molecule, let's say.
Okay, okay.
So the
And ma'am,
normal,
how does the insulin sensitivity is affected with this thing?
because they are controlling the gut pancreas axis. So in the pancreas we have three types of cell, alpha, beta, and this thing. So if beta cells is somehow altered, then no insulin will be produced. So they are directly controlling the gut pancreas axis, so through that they are controlling the insulin sensitivity. So if you are maintaining a positive gut microbiome, then this axis will be maintained positively, and the insulin will be produced sufficiently, because your pancreas is ultimately involving in the insulin production.
Okay.
So apart from that, mucosal immunity, body fat deposition will be low because of improved lipid metabolism, and a homeostasis will be there, like your body can respond towards different stimulus very quickly. Homeostasis will be maintained means uh, whatever be the condition outside of our body will maintain the homeostasis, like the temperature shift. Our body will maintain whatever it is required for a normal well-being. So homeostasis will be increased. So everything is related to the healthy gut microbiome composition. Now, another one. What are the metabolites produced by the gut microbiota? So now we can see what they synthesize, that ultimately helps us to ease the process of digestion or the process of normal metabolic machinery of our body. So now you can see the first metabolite is bile acid metabolites, including the deoxycholic acid DCA and lithocholic acid, that is LCA. So all these things are helping us to regulate our whole what to say digestive system functions, like they regulate the bile acid, cholesterol, lipid, glucose, energy metabolism, show antimicrobial effects, and activate host nuclear receptor and cell signaling pathways. So now this point, uh, this you can understand through this example. Suppose we are having we are having a cell. This is a cell. This is nucleus, and this is producing some effector molecules to its surrounding cell. So suppose this is a cell, this is the nucleus, and to sense those effector molecule, it should have a receptor thing, just the lock and key hypothesis. The key cannot sustain its life without the lock, and lock cannot sustain its life without the key. So likewise, without having a receptor, where these effector molecule will bind, so the receptor should be complementary. Complementary. So suppose this cell is the producer and this cell is the, this cell will be the responder. This will respond after binding of this effector molecule. So let's suppose there is some problem in this receptor and this is like this. So how this effector molecule will bind to this cell? So this gut microbiota, what they will do, they will help to ensure the proper cell signaling pathways, like the GPCR, the RTKs, and each and every pathways they take care of. They will help this effector molecule to bind directly with the receptor, and after the binding of the effector molecule to the receptor, the signaling cascade starts. They initiate their signaling pathway. They synthesizes the secondary messengers, and after that everything will be sorted. So this bacteria or the gut microbiome microbial diversity helps to ensure the proper cell signaling pathway, because if one cell in our body is unable to communicate with the second one, then there may be some sort of problem, because in our body there are trillions of cell and they all function together in a very coordinated way. But if somehow you are providing such condition that is disrupting the coordination between those cells, then you are in a problem. So that is ensured by these gut microbiome microorganism, that they ensure the proper cell signaling pathways. Now they also synthesizes short chain fatty acids. So this you can write it down or take screenshot, or you will get this notes also. What are the exact molecules they are synthesizing to ease the process of digestion, to ease the process of human metabolism and different things. So they are also synthesizing short chain fatty acid metabolites such as propionate and butyrate to regulate food intake and insulin secretion. Also aids in maintaining body weight. Obviously, if your insulin is fine, then there will be no fat storage, there will be no increased amount of blood glucose in your blood. So everything will be maintained in a homeostasis. Now also they synthesizes branched chain fatty acids, including the isobutyrate and isovalerate. So histone deacetylase inhibition increase histone acetylation, because histone acetylation and deacetylation is related to what? It is related to the gene silencing. So histone acetylation and everything is related to the gene regulation. So when we have to switch off the gene, when we have to switch on the gene, all it depends on what kind of function we want. Because if a gene is constitutively expressed, that is, it is in the on condition always. So what it will do, it will just harvest all the energy of the body into that, it will drain all the energy. So histone acetylation is a post translational modification where we add the acetyl group. So all these things are mediated by so many different kind of enzymes like histone acetyl transferases, and these are the crucial gene regulation part which affects the chromatin structure and the gene expression. So each and every prospect is controlled or regulated by the gut microbiome. They also synthesizes indole derivatives including indoxyl sulfate and also the IPA, that is indole 3 propionic acid. So IPA do what? It exhibit neuroprotective effects. See, you can write it down in your copy that which molecule exhibit neuroprotective effect, which molecule produce cell signaling pathway. So this IPA exhibits the neuroprotective effect, which act as also a antioxidant. It will quench the reactive oxygen species, and also it will regulate the intestinal barrier function. Endoxyl sulfate is a uremic toxin that accumulates in the blood of individual with impaired excretion system. So all these things are important. Now I was talking about the lipopolysaccharide. So this is their product, lipopolysaccharide, peptidoglycan and lipotecoic acid. So all these have epigenetic regulation of gene in colorectal cancer, modulation of chromatin structure and transcriptional activity. And apart from that, you can see so many different kind of specific molecules. Let's say vitamins including thiamine b1, riboflavin b2, niacin b3, pyridoxine b6, pantothenic b5, biotin b7, folate b11 to b9, cobalamin b12. So all these things are recommended by your doctor if you are having some sort of vitamin deficiency, because these are the commonly prescribed vitamin to the patients, because they are complaining about some hemoglobin issues, some sort of anemic disease. So they are widely recommended the cobalamin or vitamin B complex. So what they do, they help in red blood formation, DNA replication and repair, work as an enzymatic cofactor, because all this B1, B2, if you are having biochemistry in your course, you know about all these things are acting as a enzymatic cofactor for many enzymes, and they also enhances the immune functioning. Ethanol. How ethanol is produced by the bacteria? Through the fermentation. Protein fermentation metabolite may be involved in Na, FLD, progression. So all these things are very important if you talk about the biochemistry. So all these things, like whatever I have mentioned, like bile acid metabolites, the different kind of fatty acid, the short chain fatty acids, the branch chain fatty acid, the lipopolysaccharide, phenolic derivatives, and also choline metabolites, polyamines, you can categorize. Like I have written here, you can write one bile acid metabolites, an example, and what they do. Second, fatty acid, types of fatty acid may be short, unbranched, branched, lipopolysaccharide, phenolic derivatives, choline metabolites, polyamines, like spermidine. So vitamins, what are the different kind of vitamins they help to synthesize? Ethanol. So ethanol is a protein fermentation metabolite, may be involved in NAFLD progression. So NAFLD is non-alcoholic fatty liver disease, and also the H2S, hydrogen sulfide. So reduction, neutralization of the reactive oxygen species. So everything is related to the control and coordination of each and every organ of the body. So this table shows the what metabolites are secreted by the gut microbiota and their exact role in protecting our body, and through how they protect it is also mentioned. So this is very important. Now the disease associated with the gut microbiota abnormalities. So these are some of the commonly reported diseases whenever your gut microbiota will be disturbed due to so many reasons. So you can have IBS, irritable bowel syndrome, an abundance of firmicutes, and a decrease in the bacteroidetes. F by B ratio, I mentioned yesterday, is altered, then you are having IBS, type 1 diabetes, asthma, foodborne pathogen, and food poisoning. Uh, I have mentioned Clostridium. In this, they have missed Clostridium, because this is the most widely known food pathogen, Clostridium botulinum. They produce the toxic, which is a neurotoxic toxin. So Campylobacter, its species is jejuni. Jejuni. Campylobacter jejuni is one of the commonly caused food pathogen. You have read about this bacteria in the GBS, that is recently reported in Pune, Maharashtra. GBS, Guillain-Barré syndrome. This is an gastrointestinal pathogen that is commonly known as the Campylobacter jejuni. It is responsible for the gut perforation. When it is in large amount in our gut, it will perforate the intestine, and then even in severe cases, death is also reported. So this is a very, very toxic bacteria in our gut. And Shigella, they also produce toxin, Shigella toxin. So they disturb the microbiome balance, leading to dysbiosis. So everything is leading to dysbiosis, the normal disruption of the microbiome. Now, malnutrition. If there will be no microorganism in your body, then how these things will be synthesized? How these metabolites will be synthesized? So if they are not synthesized, then normal digestion of any of the food nutrient will not take place, and then no energy will be there, no fat storage, no nothing will be there. So you will be malnourished. So decreasing or missing species that either process food categories efficiently, whatever I was speaking about is written in the words. Either process food categories efficiently or produce vitamin may reduce the absorption of nutrients. If your cell are not absorbing nutrients, how they will produce energy, how they will show the ATP production? So an overabundance of enterobacteriaceae can lead to epithelial damage, diarrhea, and limited absorption of nutrients. Suppose your epithelium is choked. How it is choked? By the presence of unwanted bacteria. Then then also they will not be able to absorb the nutrient, because there is no such place. The place is blocked by whom? The bad bacteria. So this will results in no nutrient absorption. If no nutrient will be there, no energy will be there. And depression, anxiety through the gut brain axis disruption. So you will be able to know each and every term if you will see this diagram. This is self-explanatory. This one, how they control. If you are having stress and anxiety, you are having a disruption in the gut brain axis. If you are having some sort of hormonal disorder, hormonal dysregulation, then you are having disruption in the gut brain endocrine axis. So everything is related to the all these axes. So you can see here also the antagonist is Campylobacter jejuni. This is the widely reported pathogen. This is a pathogenic bacteria which commonly known to cause diarrhea, gastrointestinal perforation, and severe form of gastroenteritis. So this is a known food-borne pathogen. Wait. Now next. This is all for this unit, I think. So see, this was just the introduction, but I have not considered it as the only introduction because this is the base of the overall modules. So I guess from my side, module 1 is completed. The overview of the gut microbiome composition, function, the microbial diversity and ecosystem in the gut, factor influencing the gut microbiota. So everything is completed in a very detailed manner. Now let's have a look to the module 2. So now module 2 is all about the intermediate level. The role of gut microbiota in digestion and nutrient absorption. Now we will get to know that how specifically they, which type of bacteria is responsible for the digestion and nutrient absorption of what kind of nutrient, carbs, protein, fat, and anything. So how they digest it, how they absorb it, how the cell assimilate that absorbed nutrient, and also gut brain axis, how microbiota affects mental health. That just now we read that if a gut brain axis is disrupted, then it may result into stress, anxiety, depression, and some mental illness. So how it is related, microbiota role in immunity and inflammation, and dysbiosis. Although we have an idea about what dysbiosis is, in this unit we will go into more and more detail. We will try to reveal that what they do inside our body to maintain all those axes mentioned in that diagram. So we will start this module 2 from tomorrow, and we will solve some questions of first unit also, and then we will study this module 2. So this is all for the day. Let me stop the recording.